So very good morning and welcome you all in this e-training course on ICAR-IISR NET and ARS examination. So this is Aalok Shiv and I am working as a scientist in Division of Crop Improvement, Indian Institute of Sugarcane Research, Lucknow. In this lecture, I'll be talking about the topic given me is the metabolic processes in the plant. So, I will be discussing what are the metabolism and how this. So, basically, it is introduction. What are the different metabolic processes in the plant? And then we will be discussing the different metabolic processes like photosynthesis and respiration, and at last factors affecting and photorespiration. Metabolism, if you'll see the metabolism, so this is nothing but it is the collection of interrelated biochemical reactions that maintain the life. So metabolism composed of the anabolism and catabolism. First of all, let me tell you, this is a very general topic and we have all studied this in starting from our 11th to, still we are studying this photosynthesis and respiration. This is very general topic. So this is a kind of revision to you people. But I will be focusing, as Dr. Pawar sir has said me, to highlight those specific points which are generally asked in the competitive examination. So I will be highlighting those points. Okay. So metabolism is nothing but it is the amalgamation of the anabolism and catabolism. Anabolism, we know that the synthesis of complex molecule from the simpler ones. If we take the example of anabolism, the photosynthesis is a very good example where the carbon dioxide and hydrogen molecule join together to form glucose. What is catabolism? Catabolism is nothing but the breakdown of the complex molecule in the simpler ones. And here it releases the energy. While in case of anabolism, the energy is required. So the example of catabolism is nothing but the respiration, where this glucose molecule in the presence of oxygen broke down and we get the oxygen water molecule and the carbon dioxide. So though this catabolism and anabolism combined and it gives the metabolism. So what are the different examples of metabolism? Nothing but the photosynthesis and the respiration. So let's start with the metabolic process of the plant. The first metabolic process is photosynthesis. We all know that what is photosynthesis? It is nothing but it is the process that converts solar energy into the chemical energy, and this chemical energy is used by the biological system. That this energy is used by all of us, and it has the three major events. The first one is the sunlight is converted into the chemical energy. Next one is the water gets split into the oxygen, and third and the most important is carbon dioxide is fixed into the sugar. In competitive examination, generally they are asking what is getting fixed into the sugar. So the answer is carbon dioxide. Okay, so this is the reaction in general where the carbon dioxide along with the water in presence of sunlight, it gets converted into the glucose. Okay, and the water is the byproduct of this reaction. Next is the photosynthesis is not only carried out by the plant, but it is also carried out by the certain cyanobacteria, blue-green algae, some algae, and also the phytoplankton. So in competitive exam, they may ask if you'll have the question. So the photosynthesis carried out by the first option would be the plant. Next is cyanobacteria. Third is phytoplankton, and the last option is all of the above. So in general, those who are not aware, they will mark the plant, but cyanobacteria and phytoplankton are also doing the photosynthesis. Next, we will see what are the photoautotrophs. In science or in any subject, in concept, the terminology are very important. Here we can dissect the terminology and we can understand the meaning of it. And if you know the proper meaning of a particular term, we can clearly have the good idea of a concept. So the photoautotrophs, if we'll see, so we can dissect it into the photo, auto, and trophs. Photo means light, auto means self, and trophs means the process of making. So autotrophs means those who are able to make their own food, and photo means in the presence of light. So photoautotrophs means those individuals who are able to make their foods in the presence of light, They are called as a phototrophs, and we all know that these are the plants and certain cyanobacteria and algae, phytoplankton, which can make their own food. So these are phototrophs. And consumers such as we and herbivores, carnivores, these are depend on the product of photosynthesis. Which is prepared by the photoautotrophs. So next we will see the why the glucose is made by the plant, why not the other sugar which is prepared by the plant in photosynthesis. So we can see the glucose molecule can be broken apart for energy to power the reaction. I mean, the glucose molecule can be further broken down, and it is important for the energy. Okay, plants can also make glucose into the carbohydrate chain. This glucose is the simple sugar, and this is the smallest unit of the carbohydrate. If we have to make the further complex polysaccharide like cellulose and starch, this is can be made from the sugar. But if there will be the formation of some other intermediary or the complex product, there will be difficult to form the simple sugar and further conversion of this into other, and it will also require the energy. That's why the plants are preferring to make the glucose, and also it can convert into the complex polysaccharide like starch, and starch is the long term energy store that the plant can use later. That's why this is the reason the plants are making the glucose. Next is where this process is taking place. So this, we all know that this process is taking place into the chloroplast. So from the exam point of view, you need to remember that what are the thylakoids, what is stroma, and what is grana, and where this the individual. There are different steps and the sub processes in the photosynthesis. So you need to remember in which part of the plant and which part of the chloroplast, particularly this process is taking place. So we can see the different parts, or I can say the different compartmentation of the chloroplast. So this is the thylakoid. This is the round flattened disc. The single round flattened disc is called as the thylakoid, and the complete stack of this thylakoid, it is called as the granum. And this is the remaining portion where the liquid portion is present in the intercellular space. This is called as the stroma. So you must remember what is thylakoid, granum, and stroma, the clear difference. Okay, so this is further the subsection in the leaf. If we'll do the section of the leaf, we can have the plant cell, and under the plant cell we'll find the chloroplast, and in chloroplast we'll have the granum. Okay, so granum is nothing but the different stacks of the thylakoid, and in the thylakoid you will find the different proteins, and these proteins form the pigmentation system first and pigmentation system second, that we are going to discuss later on. And we all know about the chlorophyll, right? Chlorophyll is the green pigment, and it is most abundant, and this is generally asked in the examination. Okay. So photosynthesis is mainly divided into the two parts. We all know that, this light reaction and the dark reaction. So generally, in general, the question is asked, the light reaction, whether this light reaction occurs, whether it requires the light or not. Dark reaction is require the light or not. So this kind of question is generally asked. So light reaction is mainly, it is called as light reaction because it is the light dependent reaction. Okay, and the light dependent reaction means that is generally occur during the daytime, because the light is present only during the daytime, right? So this is dark reaction. While the dark reaction is called as the dark reaction because it is independent of the light. It does not have to do anything with the light. That's why it could either occur in day or night. That's why it is dark reaction. Okay. So the important thing from the competitive exam point of view is where does it take place? So the light reaction takes place in the thylakoid, while the dark reaction takes place in the stroma. So okay. So this is the thing you have to remember from the competitive exam point of view, that light reaction takes place in the thylakoid and the dark reaction takes place in the stroma. So in the light reaction, what actually happens is this, as I've told you earlier, we have seen, right, there are flattened disc-like structure, we call them as a thylakoid. Okay, in this thylakoid, there are different protein complexes are present, and these protein complexes form photosystem first and photosystem 2. Sometimes they are also called as a pigment system first and pigment systems 2. Okay, and these photosystem harvest the energy from photon, the energy which is coming in the form of sunlight. These photosystem, these protein harvest this energy and they convert them into the energy currency, that is ATP or NADPH. So now we'll see this light reaction in detail. So what actually happens in the light reaction, the energy absorbed by the chlorophyll during the light reaction is used to power the photosystem second that breaks the bond of water absorbed through the plant root. See what happens here is uh, here we can see the water which is absorbed by the plant root is generally dissected, or the bond between the water oxygen and the hydrogen is split into the oxygen is H plus and O2, and this energy is obtained from the sunlight. The energy which is coming from the sun, this energy is utilized and this energy is taken by the photosystem second. The protein which is present in the photosystem second, they absorb the light from the sun and they utilize this energy to break the bond between hydrogen and oxygen, the water which is absorbed by the plant root. So with the help of diagram we will see this how is this. So you can see here this photosystem second, and the energy is required, energy is absorbed, and with the help of this energy this bond between the oxygen and hydrogen is breakdown, and now this the two oxygen will join and now the oxygen molecule will form. So what will happen when this oxygen molecule will form, these oxygen molecule will now liberated through the stomata which is present on the leaf. Okay, you have seen this. Now what happens, this process will go on. Okay, when this process will go on, there will be the presence of more number of H plus ions in the thylakoid space, right? Because the oxygen will liberate and there is presence of more number of H plus ion. Now these H plus ions, what happens, there is presence of a protein in the thylakoid membrane. Okay, here you can see this is ATP maker. This enzyme is called as the ATP synthetase, right? So what happens, uh, there is presence of ADP in the cytoplasm, that is ADP, adenosine diphosphate. So what happens when there are more number of H plus ions present in the thylakoid, when it goes out of the thylakoid membrane, the protein or the enzyme which is ATP synthetase present at the membrane, it utilizes that H plus ion and it combine the inorganic phosphate. You see here, this is a individual inorganic phosphate which is present in the cytoplasm. This ATP maker, ATP synthetase enzyme, it combine this inorganic phosphate to the ADP. ADP, adenosine diphosphate. Diphosphate because you can see here there are two phosphate. Okay, that's why diphosphate, and it will, with the help of this H plus ion, this ATP synthetase, it will add this phosphorus to ADP, and now it will form the ATP. You can see here these two have been added. So now it will become the ATP, adenosine triphosphate, that is the source of energy and the energy currency what we see. So here we have seen this H plus ion which are present in the thylakoid. When they are in abundance, they are more in the quantity, they move out of the thylakoid membrane, and the protein which is, or the enzyme ATP synthase which is present at the membrane, it uses ADP and the inorganic phosphate to join them and it convert them into the ATP. Okay, so this is also happening. It happens in the photosystem second, right? Photosystem second, it convert the ADP into ATP. This is This you have to remember, okay, from an exam point of view. So in photosystem second, ADP gets converted into the ATP with the help of enzyme ATP synthetase. But in photosystem second, what happens? It converts the NADP into NADPH. This is also one of the energy currency like ATP. Okay. So two things you have to remember here. In photosystem second, ADPs get converted into the ATP, and with the help of photosystem first, the protein complexes present in the photosystem first, they convert NADP into NADPH. Okay, and this NADPH and ATP, this is required in the Kelvin cycle or the dark cycle that we are going to discuss later on. So in short, the if you see the summary of the light reaction, the photon which are absorbed by the pigments, they power the photosystem first and second. We have seen this, and in photosystem second, it splits the water molecule into uh, protons and the oxygen, which and the oxygen is expelled as a O2 gas through stomata, while the proton, that is H plus, uh, it is utilized by the ATP synthetase to make the ATP. While photosystem first, here the NADP is get converted into the NADPH. Okay, so this is a light dependent reaction, and the what is the final outcome of it? It is ATP and NADPH, and also oxygen as a byproduct. So with the help of diagram, we'll see here. So this is with the help when the light comes, you see this water molecule absorbed, this oxygen will go down, and this with the help of H, ATP synthetase will form the ADP to ATP, and with the help of PS1 will form the NADPH. NADP converted into the NADPH. Now coming to the dark reaction, what happens? In the light reaction, there is energy currency formation, ATP and NADPH. What happens in the dark reaction? The product of light reaction is used. What is the product of light reaction? ATP and NADPH. So what happens in the dark reaction? This dark reaction takes place in the stroma. This is important from the competitive exam point of view. So the Kelvin cycle or the dark reaction starts with RUBP. This is Rubisco biphosphate, bisphosphate molecule and the carbon dioxide. So this is also you have to remember from the competitive point of view. Say the dark reaction start with the RUBP, Rubisco 1 bisphosphate and the CO2. So this is the uh, starting point of the dark reaction. So an enzyme called as the Rubisco. Okay, so what happens? This enzyme Rubisco, it combines the RUBP and CO2, and it convert them into a unstable intermediate. Okay, why it is called as an intermediate? Because this is the intermediate product, and this is unstable. This is not stable. So what happens? This unstable product, this is the unstable, it broke down further and it gets converted into the two stable products. Okay, and what are these two stable products? This is nothing but the 3-PGA phosphoglyceric acid. Okay, so what happens? Okay, so this is the same. What happens when we will get? So now what is with us? So we have now the 3-PGA, which are the stable product which are obtained from the combination of RUBP and CO2 in the presence of enzyme Rubisco. Okay. So after when we are getting these stable product with the help of ATP and NADPH, which we have got it from the light reaction, they convert them into the G3P. Okay, so they convert these 3-PGA phosphoglyceric acid into the G3P. And what happens when this energy from ATP and NADP is utilized? Again, this will become the byproduct. What? ADP, inorganic phosphate, and NADP. Okay, again these molecule ADP and NADP will be utilized in the light reaction to convert them into the ATP and NADPH, which is our currency, energy currency. So what happens next is these two G3P, these two molecules join and they will form a glucose molecule. That is what is our requirement. Okay. So what is happening here in the dark reaction, if you'll see from initiation, this Rubisco, RUBP and CO2 combined in the presence of a Rubisco, and they are making an unstable intermediate, which is unstable. That's why it will further break down into the stable compound PGA, okay, phosphoglyceric acid. And phosphoglyceric acid in the presence of ATP and NADPH, they convert into the G3P, where this G3P, they molecule join and they form the glucose. Okay, so this is in short the dark reaction. So in dark reaction, whatever the ATP which has been synthesized during the light reaction, it is utilized by the plant to convert PGA into G3P, and this G3P will combine and will form the glucose molecule. So what happens? Not all the G3P we have seen is are making the glucose. What happens is most of the G3P made during the Kelvin cycle are made into the RUBP, right? And the starting molecule with energy from ATP molecule, now the Kelvin cycle can start again. Okay. So what happens? Not all the here we have seen, or this G3P molecule, they will combine and they will form the glucose. So here they are saying not all the G3P molecule are get converted into the this glucose. Some G3P molecule, most of the G3P molecule, they are not combining, and they are again spent from the ATP, and so that they are again converted into the RUBP with the help of ATP, and they are forming the unstable product, and they again are giving the ADP and the PA, the phosphorus, and this ADP and the phosphorus is again utilized for the Kelvin cycle. So this is in short the summary of the okay. Okay, this is the dark reaction summary. So the dark reaction, what happens in the dark reaction? Nothing but there is formation of the glucose from the RUBP and the CO2. Okay, these are the different structure. So in short, now we'll see the summary of the photosynthesis. So plants are the producer of the biosphere, creating the oxygen and glucose needed in most organisms, right? So chloroplast is the site of photosynthesis, and particularly the light reaction takes place in the thylakoid, and the dark reaction takes place in the stroma. Okay. So the dark reaction Kelvin cycle, this is uses the ATP and NADPH to convert carbon dioxide into the sugar. And in the very first of this lecture, I have told you that CO2 is get fixed into the sugar. Okay, this is important. In light reaction, what there is generation of ATP and NADPH energy. Okay, energy currency. In the dark reaction, this ATP and NADPH is required to convert the CO2 into the sugar. Okay, so this is in general the photosynthesis. The next part of this, the next metabolic process is respiration. So what is respiration? So what is the significance of respiration? The energy is stored as an organic food in the plant. We have seen the energy is stored as an organic food, right? We have seen the formation of the glucose, and energy is released when this organic food is oxidized during the respiration. So in respiration, whatever the organic food, say for example the glucose which has been formed, is further oxidized so that we can have the energy. And energy is really the kinetic energy which is used by all of us to do some work. Okay, and energy is generally two types: heat energy and the chemical energy. If we see the proper definition of the respiration, it is an intracellular oxidation process. Intracellular because it is happening within a cell. Within a cell, that's why, and it is an oxidation process. Sometime in examination, they are asking whether the respiration is a process: oxidation, reduction, oxidoreduction, and none of the above. So your answer is oxidation, right? So this is an intracellular oxidation process in which the complex organic substances like glucose or something, they further broken down into the simpler substance which stepwise release of energy. So the complex process, it is oxidation process where complex substances break down into the simpler substance so that there is a release of energy. I have talked about the catabolism, right? So respiration, and I have given you the example that respiration is an catabolism process. A catabolism process. So in catabolism, what happens? It releases the energy so that that energy is utilized by the different metabolic processes. So what are the different raw materials for the respiration? It is glucose, oxygen, and what is the product? It is ATP. Okay, 38 ATP. And what is the byproduct? It is carbon dioxide. So this is the in general reaction of the respiration. The glucose which is formed during the photosynthesis with the help of carbon dioxide and enzyme, it releases the energy and also gives the byproduct in the form of water and carbon dioxide. So there are two types of respiration, aerobic respiration and anaerobic respiration. And this is important from the competitive exam point of view that in aerobic respiration, there is a complete oxidation of the glucose, right? Here you can see the oxidation of glucose with the help of atmospheric oxygen. And you see there is a formation of creation of 38 ATP. While in case of anaerobic respiration, it does not require, or this happens in the absence of oxygen, and there is partial oxidation. That's why only two ATPs are formed. So you have to remember two things. In aerobic respiration, there is a complete oxidation of the glucose. That's why there is a formation of or creation of 38 ATP. While in case of anaerobic respiration, there is a partial oxidation or incomplete oxidation. That's why only two ATPs are generated. So this is the difference between aerobic respiration and anaerobic respiration. If you see the aerobic respiration, it requires the molecular oxygen, while in case of anaerobic, it does not. Here, respiratory substrate is fully oxidized. That's why we have seen in the previous slide, right? It is fully oxidized, while in case of anaerobic, it is partially. The end product is CO2 and H2O, while here the end product are ethyl alcohol and CO2. This is also important from the exam point of view. What is the end product of ethyl alcohol? Okay, so ethyl alcohol is the end product of anaerobic respiration. And anaerobic respiration, we'll see further, it is also utilized in the fermentation process, which is the basis of various commercial process. So the important point here is the 38 molecule with the help in aerobic respiration, there is a complete oxidation, and therefore the formation of 38 ATP, while here it is the substrate is incompletely or the partially oxidized. That's why only 2 ATP. And here electron transport chain is involved in case of aerobic respiration. This we will see what is electron transport chain, ETS system, while here it is ETC is not required. So here the 2 to 3 things are important from the exam point of view, that is uh, end products, number of ATP are formed, and electron transport system. So this is the respiratory substrate. What are the respiratory substrate? The initial substrate which are further broken down to produce the energy. So these are carbohydrate, fats, and protein. So this is important from exam point of view, and the sequence you have to remember. In respiration, first of all, carbohydrates are utilized, okay, and after that, fats are utilized, and at last, proteins are utilized, okay, and that's why they are the respiratory substrate under starvation. So you have to remember the sequences. First the carbohydrate, then fat, and then protein. These are the sequence of using as a respiratory substrate. So ATP, we have seen ATP is adenosine triphosphate. It is the energy currency for us, and it stores the biologically usable form of energy. So when how energy is released, when this energy currency ATP, it is hydrolyzed. Okay, so when ATP it gets hydrolyzed, it produces the 7.3 kilocalorie energy. So this is also important. one ATP molecules upon its hydrolyzation, it produces the 7.3 kilo energy. And this we have seen earlier in the light reaction. ADP in the presence of inorganic phosphate with the help of enzyme ATP synthetase, it convert the ADP into ATP. Okay, so this you have to remember. From a hydrolysis of 1 ATP, there will be production of 7.3 kilocalorie energy. And this is the structure of mitochondria. This is outer membrane, inner membrane, the crusty. Generally in the examination they are asking what are the crusties, what are the oxysomes, and which reactions are taking place in the which compartment of which organelle. Okay, so you can prepare a short notes like which reaction is taking place in the which component or which compartment of which organelle. So there are two types of respiration: aerobic respiration and the anaerobic respiration. So there are four steps in aerobic respiration. First one is glycolysis, next is oxidative carboxylation, next is Krebs cycle, and electron transport system. So glycolysis is nothing but it is the sequence of reaction in which glucose is broke down into the two molecule of pyruvic acid. Okay. So from the exam point of view, you have to remember in glycolysis, the glucose is gets converted into or broke down into the two molecule of pyruvic acid. So in exam, they will ask what is the final product or what is the product formed during the glycolysis. So the answer is two molecule of pyruvic acid. This is also called as the EMP pathway, and this is because this is discovered by the three scientists Abedin, Meyerhoff, and Parnas. Okay, this is the first step in the breakdown of the glucose. Okay, and it does not require the presence of oxygen, and there is no output of carbon dioxide. Okay, so in glycolysis, this is the first step of aerobic respiration where the glucose gets converted into the two molecule of pyruvic acid, and pyruvic acid is a 3-carbon compound. Okay, so this glucose of 6 carbon, and in glycolysis it further broke down into the two molecule of pyruvic acid. Okay, so this you have to remember. This is the process of glycolysis cycle, where it is mainly three. It gets completed into the three steps: activation, cleavage, and oxidation. Okay, this we are going to discuss. So these are the different steps. Nothing but you must have studied in your textbook. You have seen in glycolysis there are different steps. So these are the different steps written there. That glucose is converted into the glucose 6 phosphate and all. So you do not need to remember each and everything from the exam point of view here. The very good strategy here to prepare, and what point you have to remember, you just see the previous year question paper, and you just try to attempt the objective questions which are given in different competitive books, so you'll have the idea that what kind of questions they are asking generally. So though I am highlighting some of the important points, but you also refer to those objective books, and again when you're going to see or revise what you have studied, so you will be able to focus more on what is important from the exam point of view. So these are the different steps. Okay, and the last step is the ATP synthesis. Okay, and the formation of the pyruvic acid. So this is the net reaction of the glycolysis. Here we can see this glucose in the presence of ADP and NAD. It gets converted into the pyruvic acid, and there is a release of 2 ATP and NADPH. This you have to remember. The net reaction, the net gain in the glycolysis is 8 ATP, because here 6 ATP is coming from the NADH2 and 4 from the 4 ATP directly forming in this process, and 2 ATP are utilized. That's why net gain is 8 ATP. The next step in the aerobic respiration is acetylation. So what happens in the acetylation? The pyruvic acid which is formed in the glycolic acid, it's get converted into the acetyl coenzyme A. Okay, so this is acetylation. Just remember what is happening in the process and the number of ATP are formed. The next is, and very important, is the Krebs cycle. The next step in the aerobic respiration. It is also called as a TCA or citric acid cycle. So what happens here is the complete oxidation of pyruvic acid into the CO2 and H2O, and there is a release of energy. And generally, they're asking who has discovered the Krebs cycle or TCA cycle. So hence Krebs is the scientist who discovered the sequence of this reaction. Okay, and it takes place in the matrix of the mitochondria. This is very important. Generally, asking in the exam where Krebs cycle is taking place. So it is taking place in the matrix of mitochondria. So this is the different steps of Krebs cycle. This is the Krebs cycle. So starting with the pyruvate, and ultimately you'll have the water and carbon dioxide here. Next and the last step of aerobic respiration is electron transport system. This is the final step, and the most ATP and metabolic water generated in the system. Okay, and this is located in the inner mitochondrial membrane. Christie and oxygen we have seen in the Christie will find the short structures. These are called as oxysomes, and here this electron transport system takes place. And we will see this. This is in ETS. This NADH pass through the complex 1 and succinate through the complex 2, and they will join at the UQ. So generally this question is also asked that complex 1 and complex 2 are joined at which, so they are joining at UQ. Okay, so this is at last, and finally the oxygen is produced in electron transport system, and this is how metabolic water is formed. This is important comparison here. We have seen in this aerobic respiration, total 38 ATP is formed, right, from the combustion of one molecule of glucose. And in glycolysis, there is formation of 8 ATP, in acetylation 6 ATP, and in Krebs cycle 24 ATP. Okay, so total 38 ATP. This you have to remember. This they are going to ask in the examination from the glycolysis. The net gain in the ATP is, and we can say 8 ATP in acetylation, 6 in Krebs cycle 24. So from the complete oxidation of the one molecule of the glucose, we are getting the 38 ATP. This we have also studied in the comparison. So what is the significance of aerobic respiration? The one glucose molecule produces the 38 ATP. We have seen just, and the glucose molecule consists of 686 kilocalories. So this is also important, right? One glucose molecule is generally giving the 686 kilocalorie. But if you'll see the 38 into 7.3, why 7.3? Because one ATP is equal to 7.3 kilocalorie. We have seen, and this is only is 277.4 kilocalorie. What happened to the 686? Where is the rest? This remaining energy is lost as the heat energy. Okay, and only that's why the efficiency of aerobic respiration, we can say it is only 40%. 277.4 divided by 686. So this is 40%. Here some of the facts you have to remember is one glucose molecule produces the 38 ATP in the aerobic respiration. Generally, the glucose molecule consists of 686 kilocalorie energy, but only 277.4 kilocalorie energy is produced because the rest is lost as the heat energy. The next is anaerobic respiration. Anaerobic respiration is nothing but it is partial or incomplete oxidation of the organic food in the absence of atmospheric oxygen, right? So, in microorganism, it is also called as the fermentation. No gaseous exchange because there is no intake of O2P, O2 and CO2, and there is formation of only two ATP molecules. So there are three steps: glycolysis, decarboxylation, reduction. The glycolysis is same as in case of aerobic respiration. Here we can see the glucose molecule with the help of ADP and NADP, it get converted into the glycol, uh, and there is generation of 2 ATP and 2 NADH. Decarboxylation, here the pyruvic acid gets converted into the acetaldehyde with the help of enzyme pyruvate decarboxylase. So from the exam point of view, you just have to remember what gets converted, what is the initial product and what is the final product. I mean, what is the starting point? So, for example, here in decarboxylation, it is pyruvic acid. What is the final product? It is acetaldehyde, and with the help of enzyme. So, last step is the reduction. In reduction, the acetaldehyde it's get converted into the ethyl alcohol, and this is the final product of the anaerobic respiration. Okay, with the help of enzyme, alcohol dehydrogenase. So why there are two ATP? So here we can see in the glycolysis there is formation of two ATP and two NADH, right? But here you can see in reduction this two NADH is used. That's why only two ATP is remaining. Okay. What is the significance of respiration? So it releases the energy, and this energy is utilized by all of us to perform a particular process, and there is a formation synthesis of ATP. There is a stepwise release of energy, and this energy is required for the growth and development, and this energy is required for the biosynthesis, and there is a balance of CO2, carbon dioxide and oxygen, and this anaerobic respiration is utilized in commercial process like in various commercial platform for the formation of maybe cheese or various things, also in a beverage industry and everything. So this is it. Thank you so much, one and all. If you have any query and anything, please, you are welcome. Thank you so much.
Okay. From participant, you can discuss about this topic if you have any query or doubt. From participant.
Okay, even if you have no query at this point of time, and if you don't want to ask, sir can share my number with the students. I may guide them for the future, preparation of this exam, JRF.
Yes.
So you can please share my number.
Yes, I will share your number in group also, sir. So definitely, if they have any queries or doubts, so they may contact you in future.
Okay.
And, but can you give some suggestion? So how to prepare for the NET ARS examination from your side?
The strategies varies according to individual, but I must say that we must prepare in advance. You know what happens generally when we were also in our undergraduation program, we are not informed by our senior that you have to prepare well in advance. We think like this is the process of 6 months or 1 year, but it is not like, because nowadays the competition is increasing in an enormous amount. So we have to, first point is we have to prepare in advance. Okay, whenever you are taking admission in the first year, as you have joined in the second year, you are in third semester, you have to decide which subject you have to take for the preparation of either JRF or SRF, or JRF and SRF. So say for example you have chosen this soil science, or say plant science. So you have to see the syllabus first. Okay, you should first meet your seniors, and you should discuss how you should proceed in this direction. What is my suggestion is that first you should take your syllabus. In syllabus you have to see the units, which are different units, and in units what are the different topics, and you have to cover those topics from the different textbook, not only single book. What happens is students take a single book and they start reading the whole book. We should not do it like this. We should take the single topic and we should read that single topic from the multiple textbook. Next, after completion of this, we have to see the previous year question paper. What kind of questions they are asking, and they are focusing majorly on which topics, so that you can focus more, or you can divert your energy more on those topics. Okay, so first of all, take your syllabus. You should study topic wise. You should see the old question paper, and then after first or second study, you should go for the multiple choice question. I mean, you should practice those questions, so you will have idea whatever you have studied, whether I mean, you're able to apply that concept or knowledge or not. This I mean, objective types questions sometimes let you indulge in the confusion. So again, you go to the concept and you see, so it will again clarify your concept. So first of all, according to the topic, you take the different textbook and make your own notes. Okay, and from the competitive point of view, say for example, a particular concept is the photosynthesis is, so what is the actual definition of photosynthesis, who has given, what are the scientists involved. So you line wise, point wise, you make the notes. Okay, and then by referring also the old question papers, and then you, I mean, solve this multiple choice question. And also always, you know, be in contact with the senior, and when you are done with your preparation, ask your senior and your professor to take the this test, mock test, so that I mean, you can prepare well for this. Thank you, sir.
And, what about ARS, sir? ARS.
ARS is, or ARS, in ARS we have the three steps, you know. First of all, you'll have the objective. You have to clear the pre and then mains, and then you have to appear for the interview. So in pre, what we people generally do, that we do not prepare for the pre directly. You know why? Because we mainly focus on the mains. Why it is so? Because if we prepare for the mains, we will automatically going to clear the pre-examination, and the marks are decided, and your selection is mainly based on the marks which are obtained in the mains examination. If you are securing the more number of marks in mains examination, you're definitely you're going to get selected in this examination. So for the ARS, I will say the same strategy will be followed. You have to cover, you must have the previous year question paper. Okay, in plant science, if you those who are preparing don't have, they can, I mean, ask me, they can contact me. I'll try to provide them the previous year question paper. So you have the previous year question paper. In case of preparing the mains, from the each and every unit, every you should have the 2-page matter. For there are 5 marks question and a 10 marks question. So for 5 marks question, you should have the notes of 1 page. Okay, in point wise, in that you must, you know, write very practical point of view. Say for example, they have asked what is the use of male sterility, so you must be able to write 6 to 7 application of male sterility. If you want to add, if you can add some flow chart, some diagram. Okay, so this is very important. This will you know, give a good impression of your answer. Next thing is, you have to the time is very important in this exam. You have to cover, you have to write you know what, your objective and your subjective within the 3 hour of duration. And so you should know that how much time you should give to the objective, how much time you should give to the 5 marks question, and how much time you should give to the 10 marks question, and remaining you should have 10 to 15 minutes remaining, so that if you have left anything, you can cover them, you can, you know, underline the things. So this, you just have to give the good impression. First of all, you should write the, your content should be very, you know, practically oriented, and you should present it in a very good way. So first of all, you should have your notes prepared, and your notes should have each topic covered very explicitly in a very practical way. And then after this, you should, you know, appear for the your mock, you should do your writing practices. Take 5 to 10 questions every day, and try to write it within the time, whether you are able to complete those questions in the time, whether you are able to recall all the points or not. Okay, so this is how you should prepare. And for the interview, you have to study your MSc thesis, your PhD thesis, what you have done, from which university you have done MSc, then what are the important achievements of those university, who is vice chancellor, from which areas you are coming, what are the major crops. If you say, for example, you are working on the chickpea, so what is the chickpea? You should know the history, domestication of chickpea, what are the recent research going on the chickpea. So these are the different aspect or the dimension generally the panelists are asking in the ASRB during the interview. Yes, sir.
Okay, okay, sir. Thank you for sharing your views.
So if you want, sir, we can have a separate discussion on this. I mean, how to prepare for this JRF, SRF, and particularly for the ARS, sir.
Yes, yes, yes. Most of the peoples are interested in that, but they have a lot of books. But how to start their work? Means most of the people use the secondary source. Means they are
Okay.
not preparing their own notes, so that is a difficult task. Means
Yes, yes.
they can take ideas from others, but how to start, how to work, how to achieve our goals. So that is very important. So
Yes.
discussion is means most of the scientists like you, so a success story, if you can share that, how to prepare, how achieve my goal. So that is inspiration to next generation. So that
Mhm.
is very important. And this type of the lecture or training program definitely helpful for the next generation, sir. So there is
Mhm.
one question from Sakshi in chat box, sir.
Mhm.
Sir, why does ATP yield totally 36 or 38 molecules from one molecule of glucose? Sir, this no varies in difference book.
Mhm. No, no, this is actually the 38 ATP. You just remember this. This is 38 ATP, no 36. You can, I mean, refer the any book of plant physiology, you will find it. This is 38 ATP.
So, okay. This is a little bit confusing for it. That's why she
Mm,
asked you. Okay, okay, no issue. But
mm.
I will share contact number of Dr. Aalok sir. So then definitely you can contact him and
Yes.
discuss with your topic if you are facing some problems.
Mm,
So you can clear your doubt, and definitely this training program and lecture will helpful for you people, and this is only for you. So definitely a number of people will qualify the NET or ARS examination in future also. So in this year, sir, more than 100 people qualified NET examination from our postgraduate institute.
wow, so that's very good.
And among the list, 8 to 9 people appearing for main examination of ARS.
Yeah,
This
that's
is the
very
good.
good.
And this number also
Yes.
increase next year also. So that's why we are conducting this type of the training program, and most of the people like you are helping to our
This
students.
is our fortune, sir. I mean,
Uh,
we are also the product of same
yes,
university. If
yes, yes, yes,
people are not going to contribute there, then who is going to contribute? This
yes,
is our
yes,
fortune. I
yes,
mean,
yes.
we must do it. We are compliant to do it, sir.
Yes, yes, yes. You are a senior of that student, so that's why
Yes,
they will
yes.
interact with you, and definitely.
Anytime in future, sir. Anytime in future, you ask me. I mean, I'm always available for my university, sir.
Yes, 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 yes. We are expecting from you
Yes.
also. In future,
Thank
we will arrange this offline training program also, or the any lecture of series or workshop. So
you. Yes.
definitely, I will invite you, sir.
Thank you so much. Thank you so much. I would love to do it.
Yes, 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 yes. Okay. So thank you very much, sir, for accepted our invitation and deliver such a nice lecture on the this plant metabolic pathways. And on behalf of Postgraduate Institute Mahatma Phule Krishi Vidyapith Rahuri, we are very much thankful to you.
Thank
Uh, thank you very much.
Thank you so much. Thank you so much to all the students. Thank you
Yes,
so
yes,
much to all the organizers, and thank you so much to Dr. Bhale Rao, sir. Thank
yes,
you.
yes. Okay, sir. Thank you.
Thank you.
Okay, sir. Good day.
Good day. Have a good day.
Have a good day, sir. Okay, bye.
Yes. Yes.